Good morning, everyone. I am Shrikrishna P. Desai, PhD scholar from the Department of Genetics and Plant Breeding. I'm working on development of MAGIC population and tagging of resistance to major diseases in maize for my PhD work. As we all know that maize is one of the most versatile emerging crop, having wide adaptability, and globally maize is known as queen of cereals because it has highest genetic yield potential among cereals. Despite its highest yield potential, maize has got constraints for various diseases ranging from seed rots to various foliar diseases like Turcicum leaf blight. The percent yield losses due to diseases was ranging from up to global level at 9% and up to 40% in India. So there's a need of development and deployment of hybrids. Resistance to major diseases is widely considered as a most economic and sustainable and eco-friendly means of mitigating the losses due to diseases. So with the advent of DNA marker system, we are pretty much easier to identify genomic regions. So the DNA markers linked to the genomic regions controlling major diseases need to be identified for aiding marker-arrested selections. Conventionally, biparental mapping populations have been used, which has serious limitations with respect to the diversity and as well as recombination events. In order to overcome this, multi-parent advanced generation intercross have been used. The main advantage is to promote intercrossing and shuffling of the genome and segregation of the population for multiple QTLs, as well as increased recombination and mapping resolution. With this background, we have initially started our material with 22 genotypes having different responses to various diseases for Turcicum leaf blight, downy mildew, and polysorrow rust. In order to sort the things, or in order to initiate the MAGIC population project, we employed 192 SSR markers for the diversity study in order to sort the things from 22 parents to 8 parents. Based on the dendrogram constructions, we have selected 8 parents by considering both the diversity as well as disease responses for various diseases. So these are all the 8 parents that we have selected for MAGIC population development. So the crossing scheme was like this, and initially we crossed 8 parents in a half diallele fashion to get 28 direct crosses, and further 14 double cross hybrids were identified, were obtained, and from the double cross 7 8-way cross were obtained where genomes are reshuffled from the 8 parents, and further they carried on year to row method and their self to obtain till F3 progenies. At the F3 progenies, total we have generated 7 populations. Out of 7, we are subjecting one of the population with the 295 F3 progenies with the Turcicum leaf blight caused by Exorhilum turcicum for using artificial inoculation technique. And the disease reaction was taken based on the disease reaction scales from 1 to 9. And these are all the phenotypic results, phenotypic evaluation results, what we have obtained. These are the CM202, which we have used as a susceptible check, in which all the CM202 lines were completely infected with the Exorhilum turcicum. And this is the picture showing F3 progeny showing susceptibility against TLB, and this is the picture showing resistance against Turcicum leaf blight. So this is a summary of the descriptive statistic of our phenotypic evaluation, and the mean percent disease incidence was found to be 42.40% with a negative kurtosis as well as positive skewness. And this is a frequency distribution curve which we are getting for phenotypic evaluation. And the main outcome of this study is that it involves the dominance-based complementary interactions. Apart from phenotyping, we are also carrying out the genotyping for the founder parents to identify the polymorphic markers. We are using the SSR markers from 384 genomic databases, and out of 72 SSR marker tried so far, 40 were found to the polymorphic. As you can see here, SSR marker UMC 1461 was found to be monomorphic, and MMC 0041 is showing polymorphism for the founder parents. And further work planned is that during the month of this July, the progenies were screened once again for the TLB. So based on the 2 phenotypic data and the 1 genotypic data, based on the phenotypic data and genotypic data, genome association studies have been carried out to identify the SSR markers linked to the QTL. So once we identify the SSR markers at the F3 generation, so the further generation, we are planning for multi-parent advanced generation recurrent selection so that accumulation of the QTLs is possible, and simultaneously they are self to obtain RILs. When they attain RILs, when they attain homozygosity, they are subjected to genotyping by sequencing, and SNPs were identified and simultaneously screened for Turcicum leaf blight, sorghum downy mildew, and polysorrow rust as well. So based on this phenotypic SNP data and the genotypic SNP data and the phenotypic data, we are going to fine map the QTLs as well. Thank you so much.